Hello everyone, I'm Kalaivani from SNS College of Pharmacy and Health Sciences and working as an assistant professor. So welcome back to another session on pharmacognosy and phytochemistry. In our previous lecture, we discussed flavonoids, those beautiful plant pigments with diverse biological activities. Today, let's explore another fascinating and highly significant group of plant constituents, the glycosides. These compounds are present in a wide variety of medicinal plants and play a major role in their therapeutic effects. So let's understand what glycosides are, their structure, classification, sources, tests, and pharmacological importance. Glycosides are organic compounds in which a sugar moiety, that is glycone, is chemically bound to a non-moiety, non-sugar moiety, aglycone or genin through a glycosidic bond. In simple words, you can think of glycosides as a sugar-linked bioactive molecules. When they are hydrolyzed, they yield one or more sugars and a non-sugar part that usually produces the pharmacological effect. So, let's look at their structure in a simple way. A glycoside, that is equal to sugar glycone plus non-sugar aglycone. The linkage between them is through an oxygen O-glycoside, nitrogen N-glycoside and sulfur S-glycoside or carbon C-glycoside atom. The aglycone part determines the pharmacological action while the glycone part affects the solubility and absorption of the compound. For example, in Digoxin, the sugar part helps in absorption, whereas the aglycone provides the cardiac effect. So now glycosides are classified in several ways. The most common one is based on the nature of the glycone, that is, nature of the aglycone, non-sugar part. So, types of the glycosides: cardiac glycosides, anthraquinone glycosides, cyanogenic glycosides, saponin glycosides, phenolic glycosides, flavonoid glycosides, coumarin glycosides, and isothiocyanate glycosides. So, if cardiac glycosides, the aglycone nature is steroidal nucleus, and the example digoxin, and the source is a digitalis purpura, therapeutic action used in heart failure. Anthraquinone glycosides, anthracene derivatives, senosides, sena aloe, and the therapeutic action is laxative. Coming to the cyanogenic glycosides, the aglycone nature is cyanohydrin group. An example is amygdalin, and source is bitter almonds, linum, and therapeutic action release of HCN, that is toxic. And saponin glycosides, the aglycone nature is steroid triterpenoid nucleose. An example is diosine, glamazine, and source is Dioscorea and licorice, and therapeutic action is expectorant and anti-inflammatory. Phenolic glycosides, the aglycone nature is simple phenol or alcohol, and the example is salicin, and sources is Salix alba, that is willow bark, and therapeutic action is analgesic and antipyronic. Coming to the flavonoid glycosides, the aglycone part is nothing but flavonucleus, and examples is rutin hesperidin, and sources citrus fruits tea, and therapeutic action capillary strengthening. Coumarin glycosides, coumarin nucleus is an aglycone nature, and Ambelliferone is an example, and source is from tonka bean, and the therapeutic action is anticoagulant. Isothiocyanate glycosides, sulfur linkage is aglycone nature, and synergenin is example, and mustard seeds is the source, and counter irritant is the therapeutic action. As you can see, glycosides are extremely diverse. Each class has its own characteristic structure and therapeutic effect. Glycosides are widely distributed in plants and sometimes in microorganisms. Common examples include digitalis purpura, foxglove, cardiac glycosides, senna and aloe, anthraquinone glycosides, licorine, saponin glycosides, and willow bark phenolic glycosides, mustard seeds isothiocyanate glycosides. So, whenever you study a plant extract, glycosides are one of the most pharmacological active constituents to look for. Biosynthesis. In plants, glycosides are synthesized through glycosylation reactions where sugar molecules such as glucose or rhamnose are enzymatically linked to an active aglycone compound. This process increases the stability, water solubility, and transportability of the aglycone within the plant. From a pharmacognostic perspective, glycosides are usually colorless or crystalline solids. Their taste varies. Glycosides are bitter, saponins are sweetish, but produce a persistent froth when shaken with water. Most glycosides are soluble in water and alcohol, but insoluble in non-polar solvents like chloroform. So, coming to the chemical test for the glycosides, here are a few important identification tests for glycosides, and they are used in the lab. That is bond tracker stress. Boil sample with dilute H2SO4, extract with ether, add NH4OH. Then you can observe pink or red color in alkaline layer. Presence of anthraquinone glycosides. Killer Kilanitis, extract plus glacial acetic acid plus ferric chloride plus concentrated sulfuric acid gives reddish brown drink and the presence of cardiac glycosides. Legal stress, add sodium nitroprusside plus sodium hydroxide. You can observe pink or red color and the inference is cardiac glycosides. Foam test, shake extract with water, persistent froth, saponin glycosides present. And picric acid test, add picric acid, you can observe yellow color and it is the presence of cyanogenic glycosides. Ferrichlorate test, add ferrichlorate to the sample and you can get green or black color, then phenolic glycosides are present. Now let's talk about what makes glycosides so important, their biological and pharmacological actions. Cardiotonic strengthen cardiac contraction, digoxin, example. Laxative promote bowel movement, example senosides. Analgesic and antipyretic like salicin, precursor of aspirin. Expectorant and anti-inflammatory saponins enhance mucus secretion. Anti-cancer and immunomodulatory certain triterpenoid glycosides show cytotoxic effects. Antimicrobial, some glycosides inhibit bacterial and fungal growth. So, glycosides form the chemical basis for many important drugs as we use today. Glycosides have a wide range of application in both traditional and modern medicine. Cardiac glycosides are life-saving drugs in treating congestive heart failure and arrhythmias. Saponin glycosides are used in natural emulsifiers and vaccine adjuvants. Phenolic and coumarin glycosides serve as analgesic and anticoagulant agents. Anthraquinone glycosides act as a mild herbal laxatives. In the pharmaceutical industry, glycosides are important lead molecules for semi-synthetic drug development. For example, salicin was the natural lead for the development of aspirin. In short, glycosides bridge the gap between traditional herbal use and modern pharmacology. From an analytical point of view, chromatographic methods like TLC, HPLC, and LSMS are commonly used for detection and quantification. Spectroscopic methods are applied for estimation after hydrolysis of glycosides into their aglycone compounds. Advanced techniques like NMR and FTIR are used for structural elucidation. And even though many glycosides are beneficial, some can be highly toxic. Like for instance, cyanogenic glycosides can release hydrogen cyanide when hydrolyzed, which is poisonous. Cardiac glycosides have a narrow therapeutic index, meaning the difference between therapeutic and toxic dose is very small, so they must be very carefully handled. In recent years, researchers are focusing on biotechnological production, nanocarrier based delivery, AI assisted modeling. So, to conclude, glycosides are one of the most diverse and pharmacologically important groups of plant secondary metabolites, from heart medicines like this digoxin to natural laxatives like senna. Glycosides have shaped the traditional herbal medicine and modern pharmacotherapy. Thank you for watching today's lecture. If you found it useful, please like, share, and subscribe for more videos on pharmacognosy and phytochemistry. Thank you once again. Hello everyone, I am Kalaivani from SNS College of Pharmacy and Health Sciences.